Hey, deep divers, are you ready for like the ultimate mystery? Because today we're diving into the origin of life itself.
It doesn't get much bigger than that.
But forget that old primordial soup story.
Oh, yeah.
We're going way beyond that today into some seriously mind-blowing territory.
I love it.
Think clay crystals, genetic takeovers. We're talking a total paradigm shift.
Ooh.
So if you're ready to have your assumptions about life's beginnings completely shattered, hit that like button, subscribe, and get ready for a wild ride.
So it's true, the origins of those intricate systems we see in life today.
You mean like DNA?
Yeah, DNA, proteins, and all those complex molecules that keep things ticking.
Like a perfectly choreographed dance,
Yeah,
right?
like, really makes you wonder how it all began.
And to guide us through this uncharted territory, we're turning to Genetic Takeover.
Ah, a classic.
It's by A.G. Karen Smith.
Now, he doesn't just dip his toes into the origin of life debate.
Oh, no.
He does a full cannonball.
Makes a big splash.
Big splash, yeah.
With some radical new ideas.
For sure.
So picture the early Earth.
Okay, I'm picturing it.
Like total chaos. A swirling, bubbling soup of chemicals.
Primordial soup.
The classic theory says that life emerged from this chaos.
Like randomly molecules bumping into each other until...
boom. Life.
Life.
And experiments like the miller-urey one actually showed that some of life's building blocks,
Amino acids and things.
yeah, they could form under those conditions.
Okay.
So we've got the bricks.
The bricks, yeah. But Karen Smith points out a giant elephant in the room.
Uh-oh, what's the elephant?
Getting those basic building blocks is one thing, but to get life, you need a whole skyscraper.
Okay, so it's not just about having the bricks. It's about the blueprint, the organization.
Exactly, complete with plumbing and electricity. And a really good view.
Don't forget the view.
Those random collisions in the soup are about as likely to build a skyscraper as a chimpanzee is to write Shakespeare.
Rough comparison for the chimps.
Hey, no offense to chimps, but...
None taken. How do you go from a jumbled mess of organic molecules to something as organized and complex as DNA?
Yeah, that's the million-dollar question.
It's like trying to assemble a 10,000-piece puzzle just by shaking the box, you know?
Good luck with that.
And that's where Karen Smith comes in with this radical question. What if, and get ready for this, what if life didn't start with organic molecules at all?
Intriguing. What else is there?
What if the very first organisms were minerals?
Minerals, like rocks.
You heard me right. Rocks.
You're blowing my mind right now.
But not just any rocks. Karen Smith specifically points to clay crystals.
Clay. Like the stuff kids play with.
I know.
Yeah.
But clay crystals have some really cool properties that make them surprisingly good candidates for starting life.
Okay, I'm listening. Convince me.
First, clay crystals form super easily in water, even at normal temperatures. No need for lightning strikes or volcanic eruptions.
They just chill and assemble.
They're just happy to assemble. Second, they're incredibly diverse in their structure. They form all sorts of shapes and patterns. And here's the kicker. As they grow, they often incorporate tiny imperfections.
Tiny imperfections.
Yeah, like little errors in their otherwise repeating pattern.
Interesting.
And these errors, or defects, can actually store information, almost like a primitive form of genetic code.
Whoa, hold on. You're saying that imperfections in a rock can act like genes.
It might sound crazy, but think of it like a geode.
Okay, beautiful geo.
With all its intricate layers and patterns, those patterns aren't random. They arise from tiny impurities that get incorporated as the crystal grows.
Creating a unique fingerprint.
Exactly. Clay crystals can have similar fingerprints, encoding information in their very structure.
So we've got these clay crystals forming easily in water with unique patterns that can store information. But how does that translate into something resembling life?
Well, even simple properties of clay, like how well it sticks together,
Stickiness.
can be influenced by those tiny imperfections. And believe it or not, stickiness could be a basis for natural selection.
Survival of the stickiest.
You got it. Imagine an environment where clay particles are constantly being washed away by water.
Harsh.
Those that stick together better, maybe because of their crystal structure, are more likely to survive. And importantly, to grow. Over time, you get populations of clay crystals with specific properties that help them survive in that environment. So it's survival of the fittest, but with a mineral twist. How exactly did those first genetic materials, those crystal genes, actually store information? And how does that compare to DNA? Could you break that down for me?
That's a fantastic question, and to answer it, we need to dive even deeper into the fascinating world of crystal genetics.
All right, so we're going even deeper into crystal genes, those ancient information holders. But how can something as I don't know, seemingly simple as a crystal hold such complex information, let alone compete with, the elegance of DNA?
Well, it's all about perspective. We tend to think of information storage in, like, very digital terms, ones and zeros, the sequences in DNA. But nature is way more creative than that. Even the arrangement of atoms in a simple molecule can hold a surprising amount of information.
So it's not just the complexity of the structure, but how the building blocks are actually arranged.
Exactly. Think about polyethylene, the stuff they make plastic bags out of.
Okay, yeah. Not exactly the most glamorous building block of life, more like the stuff I accidentally melt on the stove sometimes.
Haha, true. But even polyethylene can store a code. See, a small percentage of the carbon atoms are actually a heavier isotope called carbon-13. They're like slightly heavier versions of normal carbon atoms.
Isotopes. Okay, I vaguely remember those from high school chemistry.
Now imagine where those carbon-13 atoms are placed along a polyethylene chain. Their arrangement could act as a code, similar to how bases are arranged in DNA. It's like a hidden message within the plastic itself.
So even a simple plastic molecule could be like a data storage device. That's pretty wild, like finding a secret message encoded in a grocery bag.
It shows us that information storage isn't limited to those complex biomolecules. Even simpler structures can hold a code if you know how to read it. And it's not just about the chemical composition either. The way units are arranged within a crystal structure, that can also encode information.
So like the crystal's architecture, how the atoms are organized within the whole crystal lattice.
Exactly. Consider the defects in a crystal, those irregularities that mess up the perfect order.
Glitches in the matrix.
Haha, kind of. And these glitches can be replicated as the crystal grows, their arrangement carrying information. It's like punctuation in a sentence, adding another layer of meaning.
So we have the building blocks themselves, how they're arranged, and these glitches that all contribute to the information a crystal can hold.
You got it. And that's where the incredible diversity of minerals comes in. We've been talking about clays, but there's a whole universe of microcrystalline minerals out there, each with its own properties and potential for information storage.
It's like the periodic table is this massive toolbox for life, and crystals are just one of the tools.
That's a fantastic analogy. And like any good toolbox, some tools are better for certain tasks. For a genetic material, you need something that can replicate accurately, store information reliably, and survive in the environment.
So out of all the tools in the mineral toolbox, what makes certain minerals, like clays, stand out as possible first genetic materials?
Remember those high-energy particles we talked about?
Oh.
The ones that leave those tracks in minerals like mica?
Yeah, I remember.
Those tracks are tiny and what we call metastable. They can last for incredibly long periods, even millions of years at normal temperatures.
Like a built-in time capsule in each crystal, preserving traces of its history. But how do these tracks connect to a crystal's ability to, store and replicate information?
Think of it this way. The deep inner parts of a crystal stay mostly unchanged, even if the outer structures reform or dissolve in solutions. So the information held in those defects, those tracks, remains intact.
So the crystal can change and adapt without losing that core information. Like a hard drive you can reformat, but the data's still there.
And that's what's cool about clays. With their layered structure, they can hold all sorts of ions and molecules between those layers. This leads to amazing variations in their properties.
Constantly remixing their genetic code, trying out new combinations.
Yeah.
But how do they actually replicate that information? It's not like they had tiny little photocopiers.
Now that's where things get really complex, and we have to talk about locally reversible mechanisms. Crystal growth, while in theory reversible, often goes in a specific direction under certain conditions.
So it's a path of least resistance, but one that can be influenced by stuff around it.
Right. And those external factors can include those defects, the irregularities in the crystal structure. As new molecules are added, they tend to follow the pattern of those defects.
It's like a self-perpetuating system. The defects guide the growth, and the growth preserves the defects.
Imagine a crystal with a screw dislocation. It's a kind of defect that creates a spiral step on the crystal surface. New molecules are more likely to attach to this step, keeping the spiral going. It's like a tiny winding staircase for those molecules to climb.
Wow, I'm starting to see crystals in a whole new light. They're not just static objects, but dynamic systems capable of change. But we still need to bridge that gap, right? How do we go from those crystal-based organisms to the organic molecules that make up life as we know it? It seems like a massive jump from, silicon and oxygen to carbon and hydrogen.
That's the million-dollar question. How do we bridge that gap between the inorganic and the organic? It's a story of collaboration, transition, and eventually the rise of a new kind of life based on organic chemistry. And that, my friend, is where the next part of our story begins.
All right, so we've gone from the world of crystal genes to the rise of RNA. We're trying to solve this cosmic jigsaw puzzle, but like half the pieces are just missing.
Yeah, I know.
And we're just like, ah.
Right.
But you know what?
What?
Let's start with the pieces we do have.
Okay, love that.
One thing that I noticed
Yeah.
when I was going through all these sources
Okay.
was just the crazy diversity of life on Earth.
Oh my gosh, yes.
I mean, we've got everything from microscopic bacteria
Uh-huh.
to these giant blue whales,
Right.
all doing their own thing
It's true.
in this one big ecosystem.
It's truly remarkable the variety of life forms that have evolved on this planet.
Totally.
And when you kind of zoom in at a molecular level,
Okay.
there's a striking unity to it all.
Interesting.
All life, from the simplest bacteria to the most complex animals, is built from the same basic building blocks.
Wow.
A handful of elements,
Really?
like carbon, hydrogen,
Mm-hmm.
oxygen,
Yeah.
nitrogen.
Okay. Phosphorus,
Wow.
sulfur.
Minerals are still essential in biological systems. Think of how important metal ions are, or the similarities between some minerals and the structures of biomolecules. And then there's the Formose reaction, a complex network of formaldehyde-fixing sugars that might be a relic of a primitive metabolism.
It's like finding out that every recipe in a cookbook, no matter how different the dishes are,
Uh-huh.
they all use the same, like 5 pantry staples.
Totally.
It's wild.
Yeah, so how do we go from this like handful of ingredients
Right.
to the mind-boggling complexity of a living organism?
That's the question, isn't it?
$1,000,000 question.
Yeah, for sure.
One of our sources,
Okay.
a book by A.G. Karen Smith
Okay.
called 7 Clues to the Origin of Life,
Catchy.
really dives deep into this question,
Oh.
and he focuses on a bacterium called Escherichia coli.
So E. coli.
Yeah, E. coli for sure.
E. coli, the poster child of like every microbiology textbook.
Right, exactly.
What makes it so special?
Well, E. coli,
Yeah.
despite its relatively simple structure,
Okay.
is a marvel of biological engineering to
Okay.
reproduce.
Yeah.
It has to create an almost perfect copy of itself.
So basically cloning.
Yeah, and that involves building all these molecular machines and
Right.
orchestrating this complex series of chemical reactions.
That's a lot of work.
Karen Smith compares this to like a factory
Okay.
with all these tiny robots.
Okay.
Each one is doing its own specific task
Gotcha.
with incredible precision.
That's insane.
It's amazing.
I'm starting to feel a little inadequate
Oh no.
compared to this microscopic bacteria.
You're doing great.
I'm trying.
So,
Hey, let's break down this factory analogy a little bit.
Sure.
What are the key components?
all right, so one of the sources mentions
Right.
these 2 key types of forces at play here.
Okay.
You've got covalent bonds
Uh-huh.
and weaker secondary forces.
Okay.
Covalent bonds are strong and stable connections that hold atoms together within molecules.
So like the glue?
Yeah, think of them like the steel beams holding up a skyscraper.
Oh, wow. Okay, I like that.
Yeah.
Very visual.
And then you've got these weaker secondary forces.
Okay.
And those govern how different molecules interact with each other.
So those are more like the wiring.
Yeah, like the wires and pipes that make the building functional.
Oh, so it's not just about like sticking the building blocks together.
Right?
It's about connecting them in the right way
You got it.
to create this system that actually works.
Exactly.
And all of this is happening on a scale that's just mind-bogglingly small.
It really is to get a sense of just how complex this is.
Yeah.
Karen Smith uses this thought experiment.
I love a good thought experiment.
So imagine building a model
Okay.
of an E. coli cell,
Uh-huh.
but you're using beads, and each bead represents an atom.
Whoa. Okay.
It would be a huge undertaking.
These a lot of beads.
It would fill up an entire cathedral with all these tiny beads,
Whoa.
all connected to represent each molecule.
Okay, I'm starting to get just how enormous this is.
It's huge.
But here's a question that pops into my mind.
Okay, shoot.
If life arose from these basic building blocks under the right conditions,
Yeah.
why don't we see new life spontaneously generating today?
That's a good question.
Like, why isn't there just a constant stream of like brand new organisms
Yeah.
just like popping out of puddles?
That is a crucial point.
Yeah.
The fact that we don't see spontaneous generation happening now
Yeah.
suggests that the conditions on early Earth
Okay.
were very different from what we see today.
That makes sense.
So the main scientific view
Yeah.
is that all life on Earth,
Yeah.
descended from a single common ancestor.
Like a single spark
Yes.
that ignited this whole biodiversity.
I like that metaphor.
So we're all part of this giant family tree
We are.
with roots that go back billions of years
Billions of years.
to this one single mysterious origin.
It's pretty wild.
But what was early Earth even like?
I know.
I mean, was it a raging inferno? Was it tranquil paradise?
I like that one better.
Or was it something in between?
Well, our sources kind of paint a picture of an Earth
Okay.
that was very different from what we know today.
Totally different, I bet.
So based on evidence from ancient rock, scientists think that early Earth had water, land masses, and a carbon dioxide-rich atmosphere.
Wow.
Oxygen levels were probably very low.
Making it a very inhospitable place.
It doesn't sound very nice.
Not for me.
Not for us.
No.
But it sounds like a pretty harsh environment.
It does.
So how did life even get started in such a challenging setting?
Well, that's where things get really interesting.
I love it when things get interesting.
One of our sources suggests that meteorites and comets might have played a role in delivering some of the key ingredients for life.
So those like dramatic fiery streaks across the sky.
Exactly.
They brought more than just destruction.
Apparently so.
He brought the seeds of life.
What a plot twist.
Right.
So even with these ingredients kind of delivered to our doorstep, going from the simple molecule to a fully functional, self-replicating organism.
That's a big jump.
It's a huge jump.
Yeah, it's like trying to build a smartphone by scattering a bunch of silicon chips and wires on a table and hoping they just assemble themselves.
It's not going to work.
It's not going to happen.
You're hitting on a key point.
Okay.
The sheer difficulty of building something as complex as a living cell. Even if you've got all the raw materials.
Yeah.
Karen Smith calls this the high-tech problem
of
the origin of life.
Interesting.
He argues that even with our modern techniques like molecule manipulation and self-assembly, creating something as intricate as E. coli from scratch would be a monumental challenge.
I'm starting to feel a little overwhelmed by all of this.
It's a lot to take in.
Yeah.
It is.
So is there like a simpler, more low-tech approach? Did early life take a shortcut? Or is this just like nature finding a way against all odds?
How do we go from these inert crystals to this dynamic, ever-changing world of life?
Good question.
How do we get the crystals to actually do something?
Yeah.
To interact with their environment and drive the emergence of complexity?
Well, that's where things get really exciting. Imagine a world where the genes are these clay crystals.
Replicating and evolving in response to their environment.
Karen Smith uses a thought experiment about a clay crystal. He calls them lumpy. Lumpy.
Okay, I'm intrigued. Tell me more about Lumpy.
I will. Okay, so Lumpy, as Karen Smith describes him, he's not your typical protagonist.
Not very glamorous.
No, he's a simple clay crystal, just chilling in the primordial soup.
Hanging out.
Yeah. But what makes Lumpy special is his ability to survive. These changes in the flow rate of solutions that are passing through his environment.
I see.
Maybe he's got like a particular shape or a surface texture that helps him cling to rocks.
Oh, so he doesn't get swept away.
Exactly. It's a small advantage.
Sure.
But in this game of survival, even a tiny edge can make a difference.
So we're seeing like natural selection at work here.
Exactly.
Even at this level.
Even with crystals.
Those crystals that have traits that make them more resilient or better adapted, they'll be the ones that stick around.
They'll persist.
And pass on those traits.
Exactly.
To their offspring.
In a way.
Yeah, air quotes, offspring.
It's a subtle process.
But over time, those tiny advantages add up. They can lead to more and more complex crystal structures.
And just like with DNA, these crystal genes, they can accumulate imperfections.
During replication.
Yeah, some of those imperfections.
Right.
They might be bad news. Leading to a less stable crystal.
Makes sense.
But some of them could be beneficial.
Interesting.
Give the crystal a new ability.
Oh, wow.
Or make an existing one even better.
So we're talking about evolution at the mineral level.
Yeah.
These crystals are competing. They're adapting and passing on their traits.
Just like living organisms.
But on a totally different timescale.
Right. And with a totally different set of rules.
This is blowing my mind.
It's wild.
And as these crystal genes evolve,
yeah,
they start interacting with their environment.
Yes.
In more complex ways.
One of the sources talks about an example where the crystal genes might have developed this ability
Yeah.
to bind and concentrate certain organic molecules,
Okay.
like phenols and carboxylic acids.
Okay. My brain is starting to hurt a little bit here.
It's a lot to take in.
So we've got these crystals,
Yeah.
which aren't alive in the traditional sense.
Right.
They're acting like genes. They're replicating and
Yeah.
evolving.
Evolving.
And now they're interacting with organic molecules.
It is kind of blurring the lines, isn't it?
It's a very blurry line between living and non-living.
It really is. And it shows us that the definition of life
Yeah.
is a bit fuzzy.
Especially when we're talking about the very beginning.
Exactly.
But here's where it gets even weirder.
Oh, I like weird.
As these crystal genes get more complex, they might actually create the conditions for their own successors.
So they're setting the stage.
Yeah, like the organic molecules that we know and love.
It's like they're paving the way for their own demise.
That's a wild strategy.
I know, right?
Yeah.
So self-sabotaging.
How does that even work?
Think of it like a symbiotic relationship,
Okay.
at least at first.
Okay.
These crystal genes,
Yeah.
through their interactions with the organic molecules, create this environment where those molecules can actually thrive.
Oh, so they're helping them out?
They're like setting up a buffet for the next generation of genetic material.
Wow.
So the crystals are providing the foundation, the infrastructure for this new kind of life.
But how do we get from simple organic molecules
Right.
to DNA?
Well, that's where RNA comes in.
Okay, the often forgotten cousin of DNA.
Exactly.
Didn't we talk about RNA's ability to be both a carrier of genetic information
Yes.
and like a catalyst for chemical reactions?
That's what makes it a perfect candidate for this transition.
So imagine these early RNA molecules.
Yeah.
Maybe they assembled on the surfaces of the clay crystals
Okay.
and they start taking over some of the jobs of the crystal genes.
Taking over the family business.
Exactly. Maybe they replicate more efficiently.
Yeah.
Or maybe they develop new catalytic abilities, and that gives them an advantage. And as these RNA molecules get more integrated into the system, they might start to outcompete the crystal genes.
They might just take over
Good.
the reins of information storage and transmission.
It's like a changing of the
A
guard.
very slow and gradual one.
So it's not like a sudden dramatic shift.
Right.
It's more like a slow transition.
Exactly.
This is so cool.
It's a really elegant solution, isn't it?
It is.
The crystals lay the foundation.
Okay.
RNA builds on that, and then eventually DNA takes over.
That's the top dog,
It's a big boss.
leading to all the life we see today.
It's a pretty compelling story.
It is. Karen Smith proposed a concept he called genetic takeover.
Genetic takeover sounds intense.
It's the idea that organic molecules, maybe simple polymers at first, gradually started taking over the roles of information storage and replication from the clay crystals.
Like a slow and steady revolution.
Exactly.
Not a sudden coup.
Imagine these early organic polymers forming in the environment around the clay crystals. Some of them might have had structures that allowed them to, like, stick to the clay surface.
Ah, so the clay crystals act as a scaffold or a template.
Exactly. As these polymers clung to the clay, they might have started taking on some of the information encoded in the crystal structure. It's like the clay was teaching the organic molecules how to organize and store information.
A molecular mentorship program.
I like it.
But this apprenticeship would have taken time.
Oh, definitely.
But gradually, those organic polymers would become more integrated into the system, taking on some of the tasks originally performed by the clay.
So the clay crystals are the original programmers, and the organic molecules are the new software, learning how to run the show.
A fantastic analogy. And just like software can eventually become independent of the hardware, these organic polymers would eventually become more autonomous.
Capable of replicating and evolving on their own.
Exactly. But it was a gradual process, with the clay crystals playing a crucial role in guiding and shaping the development of early life.
This is absolutely fascinating, but I gotta ask, why don't we see any trace of this clay-based life today?
The mystery deepens.
If clay crystals were so important in kickstarting life, shouldn't there be some remnants?
Like molecular fossils.
Yeah. Hidden away somewhere.
That's a great question, and it gets to the heart of the genetic takeover idea. Remember, this is a process of gradual replacement. As organic molecules become more sophisticated, they essentially outcompete the clay crystals, taking over their functions.
Until the clay becomes obsolete.
Exactly.
So it's like the clay crystals laid the foundation and then stepped aside to let the organic molecules build the rest of the house.
A beautiful image.
But they're like the unsung heroes, never getting the credit they deserve.
And while we might not see clay crystals directly involved in life today, their legacy could be hidden in plain sight.
Ooh, I like that.
The way organic molecules are organized, the way they store and process information, could be a testament to the influence of those early clay-based systems.
Wow, that's an incredible thought. But where do molecules like RNA and DNA fit into all of this?
RNA and DNA, the stars of the show, at least in life as we know it today, but their entry into the story is intricately linked to this fascinating world of clay crystals and early organic polymers.
Okay, deep divers, get ready to dive even deeper. We'll explore how those early organic molecules made the leap to complexity and how RNA, with its incredible versatility, might have been the bridge between the clay-based world and the DNA protein world we see today.
It's going to be epic.
Stick with us. This is where the story gets truly mind-blowing.
Welcome back, deep divers. Last we left off, we were pondering that transition from a mineral-dominated world to the emergence of organic life.
Like witnessing the grand opening of life's theater, with those organic molecules ready to take center stage.
A grand debut indeed. But the question remains, how did these organic molecules become sophisticated enough to handle the complexities of life? Building intricate structures, orchestrating reactions, replicating themselves.
It's a tall order, even for the most talented molecular actors.
Absolutely. And that's where Karen Smith's idea of a genetic takeover becomes truly revolutionary. It wasn't a hostile takeover. It was a slow and steady process, innovation and refinement.
So the organic molecules weren't exactly storming the castle, more like patiently waiting in the wings, learning the ropes, and slowly taking over as those clay crystals gracefully retired.
Precisely. Remember those catchment areas we talked about, where certain clay structures encouraged specific organic polymers to form? Well, as time went on, these polymers became more and more integrated into the system, taking on increasingly important roles.
Okay, so picture this. A bustling marketplace, all sorts of molecular ingredients swirling around, and the clay crystals are like master chefs creating little pockets, catchment areas, where specific recipes can be followed.
A fantastic analogy. Within those culinary hotspots, certain organic polymers might have had a knack for making copies of themselves. Maybe their structure, shaped by the clay, gave them a slight advantage.
Ah, so it's a molecular copying competition. Some polymers are better at it than others, even if it's just a tiny edge.
Precisely. And even a small advantage can snowball over time. The polymers that are slightly better at replicating will gradually outnumber their less organized counterparts. Survival of the fittest, even at the molecular level.
Okay, I'm following you. We've got these early polymers replicating themselves with increasing accuracy. But they're still pretty basic. How do we get from simple chains of molecules to those intricate 3D masterpieces we see in proteins today?
A great question. The evolution from simple linear polymers to complex folded proteins is where evolution truly gets artistic. It's a story of gradual refinement, each tiny change adding to the beauty and functionality of the final masterpiece.
So it's like evolution is a sculptor, slowly chiseling away at a block of marble, revealing the hidden form within.
A perfect analogy. Instead of a chisel, evolution uses the forces of natural selection. Each small change in the structure of a protein is tested in the environment. Changes that improve its function, stability, or ability to replicate are more likely to be passed on.
So how does this chiseling, this molecular sculpting, actually happen? I can't even fold a fitted sheet properly, let alone imagine how a chain of molecules folds itself into these intricate shapes.
The secret lies in the interactions between the amino acids that make up the protein. Remember, each amino acid has a unique personality, chemically speaking.
Some like water, some hate it, some are positive, some are negative.
Right. As the protein chain grows, these interactions dictate how it folds. The water-loving amino acids, like social butterflies, want to be on the outside, near the water. The water-hating ones, like introverts, try to hide in the interior. And the positive and negative charges attract each other, while, like charges repel, just like magnets.
It's like a self-folding origami masterpiece, each fold guided by these molecular forces. Amazing. But how does a folded protein actually do something? How do we go from a beautifully folded sculpture to a molecular machine that can catalyze a reaction?
That's where the magic of molecular recognition comes in. In those intricate folds, specific arrangements of amino acids can create unique nooks and crannies, little pockets that can bind to other molecules with incredible specificity.
So certain molecules fit snugly into these protein pockets, like a key in a lock.
Exactly. And when a molecule binds to the active site of an enzyme, the stage is set for a chemical reaction. The enzyme can bring molecules together in just the right orientation or strain and twist those molecules, making them more reactive.
It's like the enzyme is a master puppeteer orchestrating the movements of these molecular puppets, making them dance and react in just the right way.
I love that. And the beauty is that the enzyme's specificity is determined by the shape and chemistry of its active site, which in turn is dictated by the sequence of those amino acids. It all comes back to the sequence, the code, written in those early polymers.
This is incredible. We've come a long way from simple self-replicating polymers to beautifully folded proteins that can catalyze reactions. But how do we go from individual protein superstars to the complex interconnected networks we see in cells?
That's where things get really exciting. As individual proteins evolve and specialize, they start to interact with each other, forming intricate networks of molecular machinery, like individual musicians coming together to form a symphony orchestra.
With the genetic information acting as the conductor, making sure all the instruments are playing in harmony.
Exactly. And as these networks become more complex, they exhibit emergent properties, capabilities that go beyond the sum of their individual parts.
Emergent properties, that sounds intriguing. What kind of capabilities are we talking about?
Think about the difference between a single-celled organism and a multicellular organism like a human. A human can do things that a single cell could only dream of. Walk, talk, think, compose symphonies. These abilities arise not from any single cell, but from the complex interactions of trillions of cells working together.
So it's not just about the individual players, but how they interact and work together as a team, like a well-coordinated dance troupe, creating something beautiful and complex through their synchronized movements.
Precisely. And this principle applies at all levels of biological organization, from molecules to cells to tissues to organisms. The emergence of complexity is one of the most fascinating aspects of evolution.
This is mind-blowing. We've witnessed the rise of self-replication polymers, the artistry of protein folding, and the emergence of interconnected molecular networks. But we're still missing a key player in the story of life. Where does DNA fit into all of this?
DNA, the enigmatic maestro of the genetic orchestra. While RNA may have taken center stage in the early act, DNA's remarkable stability and accuracy eventually earned it a starring role.
But how did DNA manage to steal the show from RNA? It's like a dramatic plot twist in the grand narrative of life.
Remember how we discussed the possibility that DNA might have started as a simple functional polymer? Perhaps a structural element? Well, imagine that in some of these early gene communities, DNA began to take on an additional role, a backup copy of the genetic information stored in the more versatile RNA.
It's like having a molecular scribe, carefully copying those precious RNA scrolls, ensuring that the information is preserved for future generations.
Exactly. And as you might imagine, DNA, with its exceptional stability, turned out to be a superb archivist. RNA, being more reactive and prone to degradation, was like a busy messenger, carrying out the day-to-day tasks of the cell, while DNA held the master blueprints, safe and secure within its double helix.
It's like having a library of genetic knowledge, carefully preserved in the vault of DNA. But how did DNA go from being a backup copy to becoming the primary genetic material? It's a remarkable transition.
The key, as always, lies in the power of natural selection. Imagine that in some lineages, the process of copying information from RNA to DNA became increasingly efficient and accurate. Those lineages would have had a significant advantage, as their genetic information would be less prone to errors and more likely to be faithfully passed on.
It's like having a high-fidelity printing press, ensuring that the message is transmitted clearly and without typos. No more scribbles on those molecular scrolls.
Precisely. And over time, those lineages would have outcompeted those that relied solely on the more error-prone RNA system. DNA, the once humble scribe, ascended to the role of primary genetic material, its stability and accuracy becoming the hallmarks of life as we know it.
This is incredible. We've witnessed the rise of organic molecules, the artistry of protein folding, the emergence of complex metabolic pathways, and the crowning of DNA as the genetic king. But there's still one crucial piece missing. How did life go from these free-floating gene communities to the beautifully compartmentalized world of cells? That's a leap of epic proportions.
It is indeed, and that's where our deep dive will continue, exploring how these remarkable molecular communities learn to build walls, establish boundaries, and create those tiny, self-contained universes we call cells. But first, let's savor the incredible journey we've taken so far. We've gone from the surprising world of clay crystals to the bustling, intricate world of organic molecules.
It's like watching a grand symphony unfold, each molecular player adding its unique sound to create an increasingly complex and beautiful melody.
And as we delve into the next chapter of this epic tale, we'll discover how those early molecular orchestras learned to build their own concert halls, creating the perfect stage for the evolution of cellular life.
Welcome back, deep divers, to our epic exploration of life's origins. We've journeyed from the surprising world of clay crystals to the bustling world of organic molecules. We've witnessed the rise of DNA as the guardian of genetic information.
But the story's not over yet.
There's one final chapter in this epic tale.
The emergence of cells.
We've got these amazing molecular players, the proteins, RNA, DNA, but they're still free agents, mingling in the primordial soup. How did they learn to build walls, establish boundaries, and create those organized self-contained units we call cells?
It's a question that's puzzled scientists for centuries. It's like asking how a pile of bricks suddenly decided to assemble itself into a house.
A huge leap in complexity.
Exactly. Cells are like miniature cities, bustling with activity, specialized compartments, intricate transport systems, a central command center directing the whole operation.
Where do you even begin to build something like that?
Remember those clay-based gene communities?
Yeah.
They weren't just passive players. They could interact with their environment, influencing the formation of mineral deposits, creating more stable microenvironments.
So they're like tiny architects, shaping their surroundings to create more hospitable conditions.
Precisely. Just like a fence can evolve into a more elaborate structure, perhaps those mineral deposits started to enclose the gene communities more completely, creating a more defined boundary.
The very first cell membranes.
Exactly. Those early membranes wouldn't have been anything fancy. Maybe simpler structures, perhaps made from fatty acids or other molecules that have both water-loving and water-repelling parts.
Amphiphilic molecules.
Yes, amphiphilic just means a molecule with one end attracted to water and another that repels it.
Crucial for forming membranes.
Right. These molecules can self-assemble into sheets, with the water-loving ends facing out and the water-repelling ends facing in.
It's like those toys where you stick magnets together to build different shapes.
Exactly. And just like those magnets can form enclosed structures, these amphiphilic molecules can spontaneously form little spheres or sacs, trapping a bit of the surrounding water and whatever molecules happen to be inside.
So we've got these simple enclosures forming around the gene communities. But how do they evolve into something more sophisticated? Something that can regulate what goes in and out? A truly controlled internal environment.
That's where natural selection comes in. Remember how we talked about those clay crystals that were stickier, that could hold on to their structure better?
Survival of the stickiest.
Exactly. The same principle applies to these early membranes.
The membranes that were better at enclosing the gene community, keeping the good stuff in and the bad stuff out, would have had an advantage.
Imagine some of these early enclosures have slightly different properties, maybe due to variations in the types of molecules making up the membrane. Some might be more porous, others might be tighter, more selective.
Like different types of filters?
Precisely. And just like a good filter, a more sophisticated membrane creates a more controlled internal environment.
This is amazing. So we've gone from simple enclosures to more selective membranes, creating a distinct internal environment. What happens next in this cellular evolution story? How do these protocells take the next step towards complexity?
Once you have a compartment, a boundary, a whole new world of possibilities opens up. Different parts of the gene community can specialize, each focusing on a particular task.
A division of labor. Different teams working together.
Exactly. This specialization allows the protocell to become more efficient, more complex, and ultimately more successful.
But how does this division of labor get organized? Who's calling the shots? Making sure everything's running smoothly?
The genetic information, now housed in DNA, steps in to play the role of conductor.
DNA is like the CEO.
Exactly. Issuing commands, making sure everything runs according to plan.
But how does it communicate?
That's where RNA comes back into the spotlight.
Versatile RNA.
DNA's instructions are transcribed into RNA messages, which then travel out to the various departments of the cell.
RNA is the messenger.
Exactly, carrying instructions from the CEO's office.
And these RNA messages are translated into proteins?
The workhorses of the cell, carrying out all the tasks.
A chain of command DNA at the top. RNA is the messenger. Proteins the workers. This is incredible. We've witnessed the birth of a cell, from humble beginnings to a complex and organized unit of life.
And it's just the beginning. Cellular life opened up a whole new world for evolution.
A Pandora's box of possibilities.
Cells could replicate, mutate, compete, leading to an explosion of diversity.
Different shapes, sizes, metabolic strategies.
Leading to the incredible array of organisms we see today.
And those early cells formed communities, interacting with each other.
The first steps towards multicellular life.
Tissues, organs, all the diversity of life on Earth.
Exactly. A story for another deep dive.
It's been an absolutely mind-blowing exploration. Karen Smith challenges us to rethink the origins of life.
He shows us how evolution can transform even the simplest materials into something extraordinary.
And his story of genetic takeover shows the power of small changes over time.
So keep that sense of wonder and curiosity.
The story of life is still unfolding.
Who knows what incredible discoveries await?
If you enjoyed this deep dive, don't forget to like and subscribe
Keep those brain cells firing!
for more fascinating explorations.
Until next time, keep those brains buzzing and never stop asking questions.